I want to talk about my work. I've been focusing on investigating cellular senescence and using image-based techniques to predict senescence in tissue and in vitro. First, a little background. I'm sure many of you know, but cellular senescence is often associated with aging, both primary signs of aging and also diseases, age-related diseases. Cellular senescence is a very interesting state, a very interesting phenotype. It often occurs in response to damage or when cells undergo excessive division. And it often produces, but not always, an inflammatory senescence profile, the senescence-associated secretory phenotype. Now, despite its great value in understanding aging and disease, it's a difficult state to pinpoint. There's no universal or exclusive markers for senescence. So one characteristic, though, of senescence, apart from other marker factors, is that there's often morphological changes associated with it. There's often an enlarged, flattened appearance in vitro, and then the nuclear membrane often becomes kind of convoluted, misshapen. And these are some examples here. So we began to wonder, can we use these morphological properties to basically build a clock or to build a technique to identify senescent cells? So we started this work in vitro on human fibroblasts, and we compared control cells also to ones that had undergone replicative senescence. They're basically aged out in cell cultures, and senescence induced by radiation, IR. And for this, we used about 70,000 control cells, 20,000 IR, and 7,000 replicative senescent cells. So the first step in this process was to actually identify the cells individually, because we wanted to look at the nuclear envelope for cells, each cell. So to do that, we used segmentation methods, in particular UNet. It's a common method using neural networks to identify regions of an image. We trained it on a relatively small number of images, and we were able to pick out the cells, which you can see in red up here. And we did this to build this much larger library of over 100,000, around 100,000 cells. So then, using this library of nuclei cutout, we started to look at the morphology, and we used deep neural networks to we trained them on these images and mapped the appearance directly to an output state, an outcome state, like senescent or not senescent. And in vitro, at least, we found very, very high accuracy, nearly 90% for each of these states. So we can identify a cell as being control, RS, or IR with 90% accuracy. And that led to a very high ROC curve, as you can see here. So then we looked at senescence at the predictor, and we looked at some of the more traditional markers that senescence is often associated with. And the images up at the top here show DAPI stain nuclei along with SA beta gal, which is a common marker, as well as P21 up at the top. And we looked at the correlation in the lower left corner. You can see the correlation. And we found very high correlation for all of these traditional markers of senescence, of SA beta gal, of P16, P21, and P53. And then we also looked and compared the predictor to some traditional morphometrics, things like area, convexity, and aspect. Convexity is essentially a measure of how twisted the nuclear envelope is. And interestingly, the correlation is quite low on most of these, except area, which just tells us that the predictor is picking up on some very, I suppose, nuanced aspects of it. So then we started to make an effort to map this to tissue. All this work so far was in vitro, and that's great. But ideally, we would like to apply this to human tissue and other tissues to measure rates of aging or related to disease. So we did some normalization and used that to build a predictor, to apply the predictor to human tissue, particularly H&E stained tissue. And as one point of evidence, there's on the lower right corner, there's a chart that shows that cells in human tissue in the dermis that are P21 positive tend to produce a much higher senescence score on average, and that applies across a range of thresholds. So then we started a study to look at some patients and try to relate the senescence predictor to health and to age. So this was a smaller study. It was 169 patients, aged range from 18 to 85. And we worked with these original biopsy images from healthy regions of tissue. And the first step in this process was to take these huge images and split them into smaller tiles or patches that we could use for processing. Once we did that, we could identify the nuclei and then make predictions on it. So what we found with this group of people was we did find a relationship with age, with real age. We saw senescence, the predicted rate of senescence increased using both our RS model and the IR model, replicative senescence and radiation again. It's a relatively weak association, but it was definitely quite significant, as you can see here. And then that leads to the question of, do people with higher predicted senescence or lower predicted senescence tend to have higher or lower rates of cancer, cardiovascular, or other diseases? And this is based on Danish medical records, and they keep they centralize the records here, and they have a very good system for doing this kind of study. So we could look at this set of patients, and we could see the relationship. And a number of interesting relationships popped out, but the one that really came to the top was cancer. That was quite significant, but we also found a number of other conditions that are often associated with senescence. So in summary of this part, we created this image-based senescence predictor that can work with DAPI stained nuclei in cell culture or in tissue, as well as H&E stained tissue like what you'd get in a clinic. We use the segmentation models to identify nuclei, and then we use the senescence predictor to predict basically a numerical score of how senescent it is. And from that we can estimate the rate of senescence for a sample. And we've applied this to humans and mice primarily. We've applied this to a fairly large number of cells types at this point, including fibroblasts, neurons, astrocytes, and different human tissue and mouse tissue, including dermis, liver, etc. And we've applied this to DAPI, H&E, and briefly bright field as well. And this work went into a Nature Aging paper, which was published in 22, I believe. So now I'm going to move on to the next part, which extends from that first story. So using that senescence predictor, we began to apply it in other scenarios. We started working with data from the Komen Tissue Bank, which is maintained by Indiana University. This is a unique study in that they take samples from healthy women, and these are no diseases, no cancer, and then they track them, and we can look at the outcomes over time. So this was a fairly large study involving 4,382 individuals. And after about an average of 5 years, they would develop cancer. We saw cancer develop in 86 of the patients. So again, we're working with these large whole slide images. This is a real sample. And we broke it down into different tissue compartments. We primarily tracked epithelia, stroma, and adipose tissue. So the first part of this was to segment based on these tissue types. We primarily segmented epithelia, which you see highlighted in green on the left, and then adipose on the right. And we also used another UNet model to identify nuclei. And by knowing the location of each nucleus, we could generate different scores per tissue type, which gave us a lot of insight into various things in this study. And we also projected some morphometrics related to the nuclei using UMAP, as you see to the right, which just shows that our segmentation was effective in identifying the different tissue compartments. So then looking at our different models, and then breaking that down by tissue, I'm showing some results here. So I didn't mention AAD, but it's another model based on a combination of drug treatments, doxorubicin, antimycin A, and ATVR. as well as the IR model that I mentioned and the replicative senescence model I've mentioned. So this is quite interesting. What we found is the AED model was quite effective, especially in fat, in reducing the risk of developing cancer. And the IR model, in contrast, was quite significant in indicating and promoting cancer, or at least corresponding to higher rates of cancer. The RS also moved with IR, but it was a bit weaker. So we found models that could both predict reduced and accelerated rates of cancer. So we also investigated this with the Gale score. The Gale score is essentially the gold standard for estimating risk in individuals. It's based on basic personal history and facts about individuals. And if we combine this with our senescence predictor, we see much higher rates. So we can predict, so with Gale and the AAD model, for example, we can, it predicts like 5 times the risk, up to 5 times the risk of someone without those factors. And also something similar with, 3.5 roughly for IR plus Gale. And we also found that we can combine 2 of our models. So I mentioned one of them seems to reduce the risk and others seem to increase the risk. We can combine those models, and those 2 alone show an increased risk of like 3.3. And that's quite important because it shows these models are picking up on different aspects of senescence or different aspects that relate to risk. And this work is currently in press with the Lancet Digital Health. But I've linked to the bioRxiv paper here. And I'd like to also briefly talk about another study that we've done. So I mentioned the Komen study was based on healthy women. We've also been investigating those with benign breast disease. So every year in the United States alone, 1.5 million women undergo biopsy following mammographic abnormality. And 80% of those are negative for cancer, but they're believed to be at increased risk. And we tried to apply our methods to investigate what they say about it, if there's difference, if senescence plays a role at increasing the risk. And this is a collaboration with Einstein School of Medicine with images from Kaiser. So the chart to the left shows the risk breakdown by quartile. And so the most interesting result we found was that RS, the replicative senescence model with epithelial tissue, is quite significant and tends to increase the risk of developing cancer. Actually, and by a similar rate as what we found with Komen, about 1.7 or so. And to the right, we show how we combined multiple models, and by combining multiple models, we can get even higher rates, predicted odds of developing cancer. We found these models were most effective with 2 parts of this group, those who had epithelial hyperplasia, as well as independently as well as those who are postmenopausal. And in both of those, we saw increased numbers. And for the epithelial hyperplasia, if you combine 2 of those factors, you get around 2.5 increased risk for the odds. And in addition, we also looked at spatial distribution of senescence. So I've talked about how we can identify individual nuclei and estimate senescence scores for individual nuclei and place them in different tissue compartments. And by doing that, we can look at the spatial patterns. One very interesting property is we see very commonly a declining exponential curve like you see on the left near high-scoring nuclei. So what that means is if you have senescent cells, we see in many, many cases, the vast majority of cases, that the rate of senescence tends to be higher in that region and then decline, which fits the idea of pockets of senescence or of senescence spreading by paracrine factors, as has been hypothesized. So to the left, it's an individual sample, and then we tried to measure it with some different metrics, like using percent difference or half-life of the curve. And we found also significant results here, that independently of the rate of senescence that I talked about, the pattern of senescence affects the risk of cancer. So those who seem to have a more rapid decline in senescence near high-scoring nuclei tend to have a reduced risk of cancer, perhaps suggesting that these individuals are able to better control the rate of cancer, that their bodies are able to control that. And we combine that with the general, the average rate here, and that also increases, produces an increased odds for risk. So I've come to the end. I'd like to thank everyone, and I'm also presenting a poster, so if you want to learn more about these methods, either the general senescence predictor or its role in cancer risk prediction, I hope you'll stop by on Thursday.
Thank you, Indra. Got time for 2-3 questions. Anton, here you go.
While you were talking, I checked GitHub repositories in your papers, and actually the question is, I didn't find any actually weights published anywhere, and README seems to be empty. So how researchers can actually play with your models and reproduce it? Did you like release them separately somewhere, like Hugging Face?
Yeah, so we're not currently releasing the weights of our senescence predictor models. We are using them for some of these studies. In the case of both Komen and Einstein, the scores were generated and the results was evaluated blindly. In the case of Einstein, it was actually by a completely different group that did the analysis of our predicted scores, so we had no knowledge of the outcome. So, by way of demonstrating its independent validation. We've released some of the code, but we're currently not releasing weights.
Hi, really nice talk.
Thank you.
There are a few cell types that show sort of like phenotypic or morphological changes that are senescence-like, like activated macrophages or fibroblasts or quiescent cells. I was wondering if did you test for false positives as well, like looking at these different cell types?
Yeah, so we did a lot of different studies in vitro. We did many other related studies to try to understand that, including labeling cells, including for mixtures, including evaluating quiescence, evaluating other markers. In human tissue, it's quite difficult to actually evaluate to confirm because there's not even a standard set of accepted markers for indicating senescence. There's lots that indicate it, P21, P16, and many others, as well as SASP and other protein markers. But yeah, it's really hard to be 100% accurate, to have any kind of ground truth in human tissue.
That makes sense.
Over here, this side. And I'm wondering about just quantitatively what fraction of cells in a tissue senescent. I thought I saw in one of your graphs young tissue 10% and old tissue 15%. And intuitively it would seem that young tissue should have much less.
Right,
and the defactor should grow. So what do you think, what's the fraction of cells?
So in the case of human tissue, so what we're doing with that percent is that's an estimate, and that's based on a threshold. So we have a model that predicts senescence or relative senescence essentially. So at the current time we always have to compare it to control. We have to compare one to another so we can see elevated rates of senescence. The actual percent is arbitrary. So in that study, we essentially set the young to a certain percent, and then we looked at how much it increased for older age or the disease.
Last question here.
Congratulations. This was one of the best scientific talks I've seen in my life.
Thank you.
I would just like to ask you, do you know anything about the kinetics of the senescent cells inside those pockets and how that could promote the expansion of cancer clones? Thank you.
Yeah, we don't really. I mean, I think that's a really interesting question, and frankly, some of these results are brand new, like we've just submitted last week for the last study. These are definitely things we want to investigate further in terms of, especially with regards to spatial distribution of senescence. I think these techniques to some degree are approximate because we don't have a ground truth label for tissue, but I think they can hopefully give us a lot of insight on senescence in tissue and how it develops.
Perfect. Thank you, Indra.